Good evening. How are you? All right. Yeah. Are we excited? Yes. All right. Nothing like a free movie, right? If you'll bear with me, I'd like to introduce myself, the movie tonight, and a little bit about why we're here. So, thank you for sharing your precious time tonight and for being here. It is very wonderful to see so many of you. But I really know why you're here, and that's to see a free movie. And we'll get to that in just a minute for sure. So I couldn't be happier to welcome you to a night of what I think will be intrigue, film, curiosity, free popcorn, and science that I hope you will do two things for you tonight. The first is to open your aperture of awareness to the microbial world in ways that maybe you haven't thought about before. And the second is to elevate our town, our states, our country's conversations about science and about the microbiome in particular. So we do this in the spirit of the Penn State University motto, which is making life better. And we reaffirm that principle tonight by our science, our education, and our outreach, such as tonight. So my name is Seth Bordenstein, and I am the Huck and Dad Professor of Microbiome Sciences at Penn State. And I have the privilege of serving one of the largest and most venerable organizations in the microbiome field today, and that is the Microbiome Center at Penn State in the Huck Institutes of the Life Sciences. So the center is composed of over 500 members, from students to faculty. We have about 110 faculty in our center, which makes it one of the largest biological centers on the Penn State campus. And our North Star mission, what we look out to and what we do every day and every year, is to optimize, accelerate, and disseminate both principles and applications on the microbiome sciences. So it's in that spirit that we convene tonight at a unique time in the life sciences, a revolution that some have argued is one of the greatest shakeups of biology since Charles Darwin. Microbes were once a fringe discipline, thought to be largely germs and pathogens, but at this moment in the life sciences, at this inflection point, arguably, we now know that microbes are the base of the biosphere, and they do far more good than harm. And their universality across the globe and in our bodies can reorient our perspectives and our solutions for the next century that we'll have to deal with a warming climate, food insecurity, the rise of chronic diseases, and an increasingly interconnected human world. New data and thought frameworks on the microbiome are even asking many of us in science and outside of science, what does it really mean to be human when we learn about how human we really are? The influences of microbes on our lives and our biosphere have never been more evident to humanity than right now as we emerge out of a pandemic. This is arguably the biggest time in the microbiome's history, because everybody knows what the pandemic virus is. But from this pandemic, there is great possibility ahead. That possibility will be realized by the development of biotherapeutic bacteria, or probiotic bacteria as you may know them, that not only may sustain and enhance human health, but also will grow better agricultural products from plants to animals and modulate our environmental microbiomes where we live, where we dwell, what microbes are in our buildings, what microbes are in our environments, and how can we optimize that. So harnessing the power of microbiomes will be arguably a multi-billion-dollar industry. It's estimated that it'll be about a $5 billion industry in the next 5 to 10 years. Multiple intergovernmental agencies and academic organizations have set priorities and investments in these areas. And Penn State's Microbiome Center is leading the way. You'll have a great chance to experience that after the film when we have our panel discussion with our microbiome scientists. So it's clear to me, given the crowd here we have tonight, that we have a very special community, that State College brings us together tonight, a culture of bonds that tie us together and reaffirm our interests in the microbiome, our investments in this area, and our intellects. So after the film ends, I'll just remind you, the screen will rise, and there'll be about a 10-minute intermission. At that point, our panel will be convening on stage on 4 chairs. And after that 10-minute intermission, please join us with your curiosities, your questions from the movie. We'll have microphones floating down the aisle, so you can just raise your hand for questions. We've also received a few questions online that we'll try and get to as well. We'll have 3 scientists from the Microbiome Center and a 4th microbiome scientist who's also the research project manager, and she will be emceeing the night for us. So with that, please enjoy the show, and please enjoy your widened aperture of awareness on the microbiome. Thank you very much.
Hello. Okay, I think we'll get started here. So welcome back, and thanks for coming out tonight and staying for our discussion section. We're gonna start handing out crapsules for you to try. No, I'm just kidding. All right, well, firstly, I just want to remind the audience that our expert scientists are PhD scientists, not medical doctors, meaning they can tell you the current knowledge of the research field, but they cannot provide medical or pharmaceutical advice to individuals. So keep that in mind. And with that, it's now my great honor to introduce our expert panel. We have, directly to my left, Dr. Jordan Bazan, Assistant Professor of Biochemistry and Molecular Biology. He received his PhD in Microbiology and Immunology at the University of Western Ontario in Canada. Following that, he was a postdoctoral fellow at the University of California, San Francisco. He joined the faculty at Penn State in 2021. He has received a number of awards for his microbiome research, including a National Institute of Health K99 Pathway to Independence Award. The Bazan Lab investigates the interplay between diet, drugs, and the gut microbiome. Next, we have Dr. Daryl Coburn. Yes. An assistant professor in the Department of Food Science. Dr. Coburn received his PhD from the University of Guelph in Canada and worked as a postdoctoral scholar at the Technical University of Denmark and the University of Michigan before coming to Penn State in 2017. He has authored 37 peer-reviewed publications, and his lab studies the interactions between our diet, the gut microbiome, and health, with a particular focus on dietary fiber and resistant starch. And all the way to the end, we have Dr. Laura Wayrich. Round of applause. Dr. Wayrich is an associate professor of anthropology and bioethics. She received her PhD in biochemistry, microbiology, and molecular biology, as well as science, technology, and society from Penn State University. Yeah. After receiving her PhD, she moved to Australia for a postdoctoral research position at the University of Adelaide. Dr. Wayrich went on to be an Australian Research Council Fellow and the Node Director for the Centre of Australian Biodiversity and Heritage. She returned to Penn State in 2019 for a faculty position. Dr. Wayrich has numerous awards and honors and was actually the first person to reconstruct the microbiome of an extinct species. The Wayrich Lab uses ancient DNA to reconstruct the evolutionary history of human microbiomes, with a focus on the oral microbiome. I'd also like to mention in our audience, we have Chad Fout, who is the president of our Macrobes for Microbes Graduate Student Club, and Naomi Huntley, our student director of our Data Analysis Working Group. Yes. And so they will be running microphones to people who have questions. So if you have a question or a comment, you can just raise your hand, and then please wait for them to bring you a mic, just so everyone can hear you well. And I'll also mention that I forgot to say who I am. So I'm Nicole Ganan, and I'm the project manager for the Microbiome Center. So with that, we can start to take questions.
This is intimidating. Okay. I was curious if there's a metabolic cost to keeping up our microbiome.
It should be just talk.
Okay.
Yep.
Maybe I can start with that one. So we do some work in our bodies to try and maintain our gut microbiome. So, we heard in the movie about breastfeeding being important for having a diverse and beneficial microbiome. Actually, one of the components of breast milk are we call them human milk oligosaccharides. So they're sugars that are present in the milk that actually the baby doesn't use directly. They're actually meant to be food for gut microbes. So we have the sort of intentionality to maintain these gut microbes. And then we have this mucus layer that coats our intestinal tract. And part of its job is to kind of keep a barrier between our microbes and our cells, but it also serves as sort of a food source for them as well. So there's sugars and proteins that kind of make that up that can be a food source for a number of gut bacteria. And then otherwise, with our diet, things like dietary fiber in our diet are really good energy sources for those bacteria. So we don't use that directly. It's one of its major roles is to feed those gut microbes.
There's also examples of this in the mouth. And so some of the bacteria that live in your mouth actually feed from the proteins in your saliva and live there. But it is quite interesting to note that across different human populations that have very different microbiomes, as you saw in the movie, that the sort of resting basal metabolic rate of those populations remains the same. And there was a really awesome talk last week in the Department of Anthropology looking at that. So despite people, for example, the Hadza, have very different gut microbiomes, living in places in Africa have very different microbiomes than us living here in industrialized countries, we still have the same overall metabolic rate. So there is some sort of balance that humans achieve with their microbes, regardless of where we're living and how we're living.
We can take our next question. I see someone in the...
What's the most powerful slash like overtaking microbe there is, good or bad?
I think I personally would say Shigella, which is like the evil cousin of E. coli. It's a very infectious and potent microbe, which, fun story, a former boss of mine accidentally infected themselves with. So you got to be careful working in the lab.
Yeah, some pathogens have ways of hijacking your immune system and using your immune system to clear out all of your gut microbes. So bugs like Salmonella can use some of those tricks and tools. I would call them pretty nasty as well.
Yeah, I think I would. I like the Shigella answer. Just to add on to it, you only need like a very few number of these bacteria to cause disease. So when we often talk about like infectious doses of like how many bacteria or of a virus that you need to cause an infection, this is like one of the lowest ones that exists.
I think about pathogens as just really good microbes for outcompeting other microbes in our bodies. They're kind of the meanest and the, I don't know, the nastiest towards other microbes in our bodies. That's the way I think about pathogens.
Let me offer a counter take if we can get contentious. I actually like to think about pathogens as being worse. microbes.
Worse.
Yeah, so hear me out on this one, though.
Okay.
So, if we really think about what's the goal of a microbe, right? If the goal of the microbe is to reproduce and pass on its DNA, what's more successful? The pathogen that affects, 0.1% of people, or is it, that particular healthy gut microbe that's in every single person in this room, right? So I think they're two different lifestyles. But sometimes I like to think almost of a pathogen as sort of an immature version of a healthy gut microbe.
Something that's needy and takes up your resources.
One thing I'd like to mention with that too is that pathogens are still considered to be part of the microbiome. They're not separate. And in some ways, you can think of the microbiome as more of a gradient of helpful to harmful, rather than separating these into pathogens and beneficials.
Yeah, I think that's a good point because, like you say, a lot of the ones that we can consider pathogens, it's only when they're at too high of a level. A lot of those things, we heard a lot about C. diff, Clostridium difficile in there. Tons of people have that naturally within their gut microbiome. It only becomes a problem when it gets to too high of levels, say when you take a course of antibiotics that eliminates a lot of the other good microbes and it can kind of take over. So that's something to really keep in mind.
Hello. So I have a question, as you sort of talking about separating a moment ago. So I'm curious about how, when you're looking at the microbiome, which itself is complex, how do you control for other potential factors that have an effect on the gut or the body, for everything from maybe steroids to hormones and other sorts of factors?
So, I think this is why we see so much animal research done in the microbiome field, because it's actually very difficult to do those studies in humans. The amount of what we would call metadata, or sort of additional information we need to collect, is really staggering to be able to control for everything. And so that's why we often turn to animal models, as you saw in the documentary, with the giving antibiotics to mice and having them become obese. And so, through these animal models, we can ask all sorts of questions, including using these mice that don't already have microbes in them, right? That sort of gives us a clean slate where we can manipulate only the microbes without any of the other factors.
Yeah, I'll add another perspective to it too. As we've done a few different human trials in my lab, and one of the ways we get around that is we kind of cheat. So we are interested in like these different fibers and resistant starches and what their effects are in the gut. So what we do is we give people a ton of them, way more than you'd normally have in your diet. And so it's enough to kind of see the signal from that, even with all that background noise of all that other stuff.
Yeah, I think it's a very complex issue. And for us, it's even compounded by time. So much of what we do in my lab is look at ancient humans where you can't go ask them 150 questions about what they ate and what they were doing. And so we have to use other proxies from the environment, from isotopes in their bones, from all sorts of things to try to infer some of the information. But in many cases, you're going to detect the biggest signals and may miss much of the smaller, often very interesting findings. And so we have to do these studies eventually in humans to move out of mouse models, but we have to think very carefully about how we design and how we block our experiments to try to reduce that variation. And our poor subjects, we ask them a bajillion questions from anything about, what they've eaten to where they grew up to the water that they drank when they were little, all sorts of questions. So it's kind of probably a statistician's nightmare, I think, most of the data sets that we have.
I think the documentary really illustrated this problem nicely with the fecal transplant trial for obesity, right? In mice, this works beautifully, right? But that trial and a couple others published over the past couple of years have been miserable failures, right? Even though on paper they should work. And I think this just reflects the complexity of the ecosystems that are actually inside humans, right? And going into this idea of fecal transplants, right? If everyone already has sort of a different starting place, can you just insert a new community on top of that? Probably not. And so how would you open up spots in that community for new microbes? Antibiotics, right? And then we get back into a circular issue. And so there's a lot of challenges, I think, as these therapies, especially FMTs, start to go into humans related to those exact problems.
Oh, son. I actually changed my question after hearing the last response, but have there been any studies where they've tried using antibiotics to kind of clear room for new microbiomes in humans? Has that had different results?
Yes, and in fact, it's really quite common, and it can be antibiotics and it can be other ways to try and deplete the gut microbiome, like using PEG and other things like that. The idea is to flush out as much of what's already there so you can replace, and that's actually a very common practice, even clinically, with FMT usage.
My team's thinking a lot about microbial transplants in the mouth and not necessarily the gut. And the mouth is kind of convenient because we can get in there physically. So we can do physical removal of the oral microbes by scraping your teeth. And then we can also do chemical treatments with antibiotics or things like chlorhexidine, other antimicrobials that can really try to sort of, nuke the existing microbes in the mouth before we might put something else in there. So we have to think about how to remove or to alter the recipient microbes to make the environment more receptive to donor organisms as we do transplantation.
Question about those kind of like all you are talking about is microbes. Use for microbes is very important, play an important role for the human being's body is kind of like the healthy. What currently we are trying to do is keep those useful microbes, and some microbes, useful microbes, is kind of like going to disappear, extinction. But currently, all you are talking about is kind of like, reduce those drug use, antibiotic, and eat healthy, and all those kind of like treatment somehow is keep those useful microbes. And I have a question about this currently, your research, especially those your kind of like microbiome, the centers. Do you have any directions for your research, kind of like, keep reproduce those useful microbes, and it's going to be a breakthrough for those research, public health. If we can find a way, especially your research, okay, all those kind of like your labs, even molecular biology, the protein biology, whatever, can find a way to reproduce those, good microbes, and or maybe they use the UI to redesign those useful microbes. It's kind of like already maybe kind of like disappeared. Do you have any of those kind of like a research direction currently?
Yeah, so I think we're all probably doing that to some extent, but I can probably start there. So, one of the questions we have in our lab is, we saw the power of fecal transplants, but then we also saw the blender and the administration, and this idea that we don't actually know what's in them. So what we're trying to do is take organisms that we can grow in the lab and rebuild a synthetic alternative. Right, and there's a lot of challenges there. How can you take microbes, these healthy microbes, and get them to coexist in a single environment? It's not a straightforward task. And how many microbes do you even need to represent a fecal sample, right? The answer in our group is 40. Maybe it needs to be more than that. We're still trying to figure that out, but that's a big part of it. And this idea too of engineering microbes. I think that's a really exciting place right now in biotechnology as well. Probiotics, or at least what's generally found in sort of over-the-counter supplements, they represent really narrow slice of diversity. Generally the lactic acid bacteria, so like Lactobacillus bifidobacteria, but that's a very small slice of what's actually in the gut microbiome. So can we reach out into the rest of diversity and get those good microbes? And then sometimes those microbes might have good traits and bad traits, right? We talk about pathogens and healthy microbes, but really there's quite a gradient in between, or sometimes it's contextual. And so a big question is, well, how can we take the good trait from this microbe, that might be a bad microbe, and then express it in a surrogate organism like a lactobacillus? And so that's another thing we and others doing, and some colleagues of mine actually in the biotech space are also doing it. It's a very exciting place right now.
Yeah, and sorry. One of the other issues too is you got to figure out what microbes you want to add in, but then you got to find a way to get them established in there too. And so that's a challenge. So kind of part of what's happening in the FMT is you're just putting in a ton of microbes in there to kind of, and usually into an environment where they're already depleted, to try and get them established in there. But what about, if you're relatively healthy and you just want to take some capsules to add some bacteria in, how effective is that going to be? A lot of times, people have looked at when you take these probiotics and then see if you can see them inside the body. So as you're taking the probiotics, you can keep detecting them in the stool samples after that. Then you stop taking it, and then after a while, you no longer see it anymore. Because it's really hard to get these other bacteria established in there. You can think of the gut as an ecosystem where there's these niches, and if a niche is occupied, it's really hard to displace that organism out of there and get a new one established in. One of the things we look at is exploiting some rare abilities that some of the microbes might have, say a certain energy source that they can use that only a very few other bacteria can use. Can that help that bacteria now get established in that environment? So, it's one of those challenges. You got to think of how you're going to get those bacteria established in there too.
Yeah, it's a great question. My lab's thinking about this a lot in the context of our evolutionary history. And so I absolutely commend the work that Gloria and others are doing to, take these microbes from, for example, indigenous populations and keep them in freezer biobanks. But those microbes are also very much adapted to people living those lifestyles. And people living in industrialized societies don't eat, for example, the same amount of fiber that people living in the jungle eat. And so it might be a little bit of a misnomer to assume that some of those microbes that do really well in people, say Venezuela in the jungle, would do well in our industrialized guts here in the United States. And so my team thinks a lot about, okay, what did industrialized guts look like before the Industrial Revolution? And are there people who live in industrialized settings, who eat industrialized lifestyles, who still then have really healthy gut microbiomes, or in our case, oral microbiomes? So who are the people who have never gone to the dentist, never brushed their teeth, and still never had a dental cavity, or never, never been in the office for periodontal disease, but still eat lots of sugar, right? Those are the people's microbiomes that we really want to study and look at. So we think a lot about our context from the world that we're currently living in, and how we might be able to leverage microbes from people who are living this current lifestyle as a way to improve our health, both by culturing those unique microbes, but also by doing similar things to what Jordan's doing by engineering microbes to have those sort of beneficial traits.
That's excellent. We can take the next question.
So I would like to bring the conversation a little bit further. So the first one would be, it was kind of illustrated in the movie earlier that for kids or people with autism, the way that they eat or behave is like totally different from a normal person. But which also brings in the idea that those are creating confounding variables in the research. So what's your opinion on research on autism and microbiome, and if possible, how we can do better in the research in this field? And then my second question would be regarding pathogens. So, what's your definition of healthy versus disease from a microbiome standpoint?
Who wants to step up to that plate?
Go for it. Yeah.
So maybe we can start with the autism question. And this is an area that I have sort of followed with some interest since it sort of came onto the scene in maybe, I don't know, like 2013, 2014. It's really tricky. Again, we go back to this idea of, we have mouse models where it seems to be really convincing, but the human data has always puzzled me a little bit more. And there's a really amazing paper that came out this last year where they really carefully controlled for the dietary preferences of autistic children. And as it turns out, a lot of the signal we see can actually be ascribed to diet rather than autism itself, although there were a couple of sort of leftover microbes that survived the correction. But again, it goes back to this idea that studies need to be really carefully controlled, and we need to get at cause and effect, right? And that's very different from correlation, which is what a lot of studies are really designed to find.
Go ahead. No.
Yeah. the autism research is I would say still very much in early days. The movie portrayed it as if it was, I think, a bit further along, but from my perspective and what I've been following is very much in line alignment with what Jordan has said. So I guess I'll move to your second question and sort of what's our definition of a diseased versus a healthy microbiome. And this is actually a really key issue in the field because there's this term called dysbiosis, and you will read it everywhere. And it just means a shift or a change in the microbiome. But what is dysbiosis in one person may be completely healthy in another. And we really don't, we understand that in some diseases better than others, but this notion that there's sort of one healthy microbiome and that there's one sort of disease state really goes out the window. Really, it's a spectrum. And depending on people's, again, evolutionary histories, their backgrounds, their lifestyles, their diets, you're going to sit on that spectrum somewhere. And so, we really have to think about almost a personalized medicine approach and how we think about the microbiome and how we think about whether or not somebody could be receptive to an FMT or, some sort of, probiotic treatment.
Yeah, and I think it's another part of the issue that you can think of is when a lot of the studies of looking at what does a healthy microbiome look like have been sort of Western based, for instance. And so, what looks good in, a US metropolitan population might be completely different than what a healthy microbiome looks like in Southeast Asia, and all these other places. And some of that might be diet and lifestyle differences between those places. Some of that might be that the bacteria that we kind of think of are the same might be different sort of strains of those bacteria in different places that have totally different traits that affect how healthy they are. So it's one of those things where we just need to take a fairly holistic view of it. And I think it's better to think of, rather than something being totally unhealthy, trying to get down to what are the particular aspects that might be contributing to a disease rather than trying to define just what a healthy microbiome is.
And to add to that, I think one of the really exciting opportunities in translational microbiome science is this idea that, the microbiome that might be good for someone with, autoimmune diseases might be the microbiome that's bad for someone with cancer, right? And this idea that we could sort of shift the microbiome into various states to sort of suit whatever the disease or illness is a very exciting opportunity. And yeah.
Hi, I have another question, and this one connects a little bit closer to food allergies. I'm in a very unique situation where I study tropical marine science, but I'm allergic to fish. And since I work with it all the time, and sushi looks amazing to me, I wanted to get your insights on the connection between food allergies and microbiomes found in the mouth or in the gut, and then what are your thoughts on this as a potential therapy? I know we talked about manipulating microbiome and the challenges associated with that, but could this be a useful therapy for people who have more severe food allergies where they might be a little bit more able to go out and try different things and live a normal life?
So the irony is I work with mice most of the day and I'm allergic to mice. So I feel your pain. Don't tell occupational health. So I think, the key with, microbes is when we start to think about the immune system, people often think, okay, there's an immune response or there isn't an immune response. The immune system's on and off, but that's really not what it is. Microbes are constantly twisting knobs. They're dialing it up, dialing it down. And by manipulating microbes, we do have this capacity to sort of turn down, the immune system's response to various allergens. Again, it's one of these areas where, we have a lot to learn, and I think the documentary covered sort of one organism that could be part of that. But again, through this idea of manipulating the immune system via the microbiome, there's a lot of exciting options.
Go ahead.
I think one of the more exciting areas, which they just briefly mentioned in the film, is pairing that sort of reintroduction of small amounts of allergen with probiotic microbes. And so there's some really amazing clinical research going on in Australia doing FMT of young children who have severe food allergies and seeing some pretty amazing results, especially with peanut allergy, and really thinking about getting kids early and trying to retrain that response using sort of an FMT plus dosage, small dosages of the allergen. And for me, that's like super exciting because I think, the sky's the limit then. How many different allergens can we sort of use in this way? But it's just early days, right? And a fecal transplant is just a replacement for not understanding the specific mechanism, right? And the specific microbes that are necessarily involved. So, I don't know, it's my hope that 20 years from now, it'll just be like, oh yeah, just take that allergen with this microbe and, 2 weeks later, you're fine for the rest of your life, right? Maybe we'll never get there, but, that's the hope and the dream that microbiome science puts in front of us. And, we'll work towards it.
Yeah, I'll add one other piece to it in that, so there's like the possibility of the microbes, sort of immunomodulating us so that we don't react the same way to that allergen. The other thing is they can potentially degrade the allergen itself. So, a lot of the times these allergens are proteins that some part of them is surviving through, and it activates our immune system, because our immune system usually is recognizing proteins when they're looking for potential pathogens in the environment to react to. And so some of these proteins are more difficult for our digestive enzymes to break down, but microbes have different sets of digestive enzymes, and sometimes they're better at breaking down some of these more difficult to degrade things. So it's possible that might help play a role in preventing or alleviating some of these allergies too.
Yeah, I just want to add to Daryl's comment, because that's an awesome one. I think one of the most exciting areas of research that's coming out in that space is thinking about lactose intolerance. And so humans, produce lactase, this enzyme that allows you to break down milk when you're small, and then in some people with particular genetic profiles, stop producing lactase later in life, which is what makes you lactose intolerant. But in fact, there are populations that exist on the planet today, including in the Tibetan Plateau, where people don't have the production of the lactase gene, but can still drink lactose. We believe it's through the microbes in their gut that are sort of providing that function to break down the lactose for us, so that it doesn't cause our bellies to hurt. And there's, a lot of pharmaceutical excitement about that, to think about the sort of probiotics that go along with that. But, that's an example of not necessarily retraining of the immune system, but really of these microbes replacing a human function that we would otherwise have.
You can take the next question.
I have related to the competition between the commensal and pathogenic bacteria. So, as we know that the commensal bacteria or the healthy bacteria are in kind of a symbiotic relationship with the host, like they utilize the nutrients from the host to survive. So, in case of a pathogenic infection, is there a way to ensure that the pathogens don't win in the competition and it's the commensal bacteria? Is there a way to ensure that the commensal bacteria thrive in such a situation?
I mean, there's no surefire way to prevent these pathogens from getting in. Part of the reason why they're prevalent pathogens is because they're really good at kind of creating space for themselves to invade into an environment. Having said that, there's all kinds of evidence that having a sort of healthy, more diverse microbiome in general often can help provide what we call colonization resistance. So preventing some of those pathogens from kind of getting established inside us. So I mentioned that issue of if we wanted to try and get healthy organisms into us, how we need to have them kind of get established in there, because there's all these niches are kind of occupied. Well, that same thing can help prevent the pathogens from getting in there too. So the pathogens got to come in there and find some way of exploiting resources to grow and then cause disease. So if you have a really robust microbiome, it makes it harder. It doesn't make it impossible usually, but it can make it harder for them to get established in there.
Yeah, and sometimes I think too the question is, well, can we tilt the favor towards the healthy microbes, right? And so that's where dietary fibers or prebiotics often sort of come into the equation. This idea of, well, can we put in a nutrient that only the healthy microbes can use, but the pathogen can't, which creates sort of a selective pressure or an advantage for them in that competition.
In the documentary, they mentioned that 75% of antibiotics are sold and fed to animals. What sort of effect is factory farming and industrialized farming having on the Western gut biome?
Yeah, so I mean, it's something that is been a real concern and has led to reductions in uses of antibiotics in animals, and so we're getting more and more towards only if there's actually an active disease in the animal giving them antibiotics. So when they showed you that pig that you could feed it antibiotics to get it grow faster, we're moving away from that here, and it's increasingly around the world too. But has some damage already been done? Probably. So the antibiotics that are approved for animals aren't the exact same ones that we give to humans usually, but they can have sort of related mechanisms of action, and sometimes if microbes become resistant to those ones, they can have cross resistance to other ones that are important in humans. And so if you think about pathogens like E. coli 0157, the main reservoir of those that we worry about is in the guts of cattle. And so if you're giving your cattle antibiotics, those E. coli can now become potentially resistant to those antibiotics over time. And now when they start causing disease in humans, you might have a multi-drug resistant E. coli infection, which is very difficult to treat.
I think about this a lot in the context of also food storage and, the types of microbes that we would have been consuming naturally on foods before they were processed and, canned and sterilized for long-term storage. And so, if you think about prior to the Industrial Revolution, prior to the canning lines existing, we were eating a lot more fruits and vegetables and food sources that weren't necessarily sterile. And so we're thinking about consuming a lot more microbes that didn't necessarily go and live in our gut, but may have had sort of passing benefits or temporary benefits for us. And so I think a lot about, how different the microbes are that we consume in the food that we eat today. And there's been some really amazing research just on apples. So apples can sit up to 2 years before people consume them as they're sort of waiting from when they're picked into when they end up in your home in industrialized settings. And there's been some really amazing research watching the microbiome of apples change over time as they sit and wait to go on grocery store shelves. And, we consume many fewer microbes and very different types of microbes in these foods that have sat around and waited for us to eat them. So, thinking about that from a broad term evolutionary context for me is quite interesting because could it be fueling some of our, fewer diversity in our microbiomes? Then maybe, and we really don't understand that process yet.
One adjacent thing to mention, because I know the work of some of our other faculty in the center, is when we are no longer giving these livestock certain antibiotics, then we don't have that growth promotion that they mentioned in the video. And so then we go back to, okay, how can we use other microbes to still get that growth promotion that we saw with antibiotics? And there are faculty affiliated with our center who are working on questions just like that too.
I'm thinking a lot about how a lot of the diseases that were mentioned, like obesity and diabetes, are also associated with socioeconomic background. And I was wondering what kind of research do you think is still needed to kind of make these therapies or prevention methods more broadly accessible and affordable for more people?
I think about this a lot, right? And I think about this especially in the context of the way race is used in microbiome sciences. The NIH still requires that we collect ethnicity or race information when we know much of what's actually being described in race is actually a social construct that's based on social, cultural, historical, and even environmental contexts. And so for us, we have to understand what's actually driving differences in the microbiome and think about that on a social science level in order to really understand how we then shape microbiomes without using sort of pharmaceuticals. So I'm part of an initiative here with Seth and others trying to really build bridges between the social sciences and the microbiome sciences so we can get better tools to discern microbiome shifts in the context of social cultural change. because right now we're sort of using really big, broad categorizations that don't get to the nitty-gritty of, okay, did you grow up in a red line district or did you not, right? And does that play into whether or not you have different microbes in your body? I think it's also really important to note that as we're developing therapies moving forward, we need to make sure that our FMTs and our probiotics work for people of all ethnicities, right, and of all backgrounds. And that's something that's not really in the conversation yet. We're still trying to figure out what even is a successful FMT, not is it successful across all populations and people of and available to all people of different socioeconomic status. And so for me, those are factors that really need to enter into the conversation and enter in now, so that as we're building these therapies in the years coming forward, that they're accessible to everybody and that they work for everybody in the same way.
Yeah, and I think too that as these things like next generation probiotics or synthetic microbial communities to replace FMT, and they kind of become established, they're potentially a fairly cheap option compared to treating the lifelong disabilities that might be associated with diabetes or obesity. So the hope would be that insurance companies would pay up for people to do that. But
and I think a big thing is it really comes back to the data and the amount of data we have. And I think being a newer field, it's not uncommon to see studies with 20 people or 100 people. But we need 1,000, 10,000, 100,000 people in our studies where we've captured this range of diversity and across all of our different metrics, and then that will give us the ability to actually tease a lot of this out.
I'll also mention that at the center level, this increasing the diversity of our samples and our knowledge about the microbiome through all different walks of life is a really high priority. And so we have been building infrastructure and collaboration groups to focus on these issues. We can take the next question.
Hi. Oh. Okay. Sorry. In the movie, it talked about how microbes can train the immune system. Is that more of an interaction of the microbe communicates somehow, or is it more just the body learns to attack bacteria instead of food?
I mean, some of both. So, as you are developing your immune system, as an infant and a small child, part of what's happening is your body is seeing what's normally around there. And so if you have this diverse microbial population in there at that time, your body sees that and recognizes that as sort of part of the normal background. But at the same time, they are producing a lot of metabolites that directly influence the immune system as well. So it is kind of both aspects of that.
Yeah, and I think it's also as well exposure to pathogens and things that may also not be your microbiome, and sort of, again, educating or training or teaching tolerance to your microbiome. Like, okay, react to that pathogen because that's really awful for you, but don't react to these. So it's a bit of that switch, and when do you turn on full force and when do you just sort of sit back and just be tolerant of what's going on in the body? Hard to explain.
Next question.
Yeah, so before the movie, like Dr. Bordenstein, and even during the movie, there was a lot of talks about clinical possibilities of the microbiome field. So I'm really curious to hear your opinion on what the future of microbiome medicine would look like, maybe some of your wildest ideas, hot takes possibly.
I'll give everybody mine, I guess. I do think that we have to be very cautious, and the movie shows one example of when the microbiome didn't work, but FMTs can be successful up to 85% of the time, but FMTs in treating things like inflammatory bowel disease are maybe 35% of the time. If you change the diet, maybe you're upwards of like 55% of the time you might get clinical remission, but they're not nearly the smoking gun 100% of the time everybody's cured, right? And so I think one of the big pushes is to increase its efficacy, probably through, working on transplantation, but also what are the prebiotics, what's the diet, what's the environmental shift, you also need to keep that. So all of that said, I really do love this field because I think there are a lot of opportunities for these, really amazing transformative therapies. And if I had a pipe dream in 30 years from now, no one would have to brush their teeth and you wouldn't get dental cavities and you all have beautiful smelling breath because we would be transplanting oral microbes into your mouth that would prevent oral disease, but also be, something that is enjoyable to you, maybe that even improved the flavor of the food that you took that day. Yeah.
You want to go, or because I was just going to quote you basically. So.
Oh, okay. I could give a spicy take, I guess. I think that the future is probably not lactic acid bacteria, which is probably mostly what you would find in probiotics today. I think they have a lot of amazing traits, but again, it's like we're trying to paint with such a narrow spectrum of the color that exists in the gut microbiome. And so I think we're going to see a lot of organisms that today are actually labeled as pathogens, but we'll have figured out that maybe they're not really pathogens, or we'll use variants of them that are no longer pathogenic to recover those sort of traits. And I think between that and genetic engineering of microbes, we'll see a really big shift in what's available.
Yeah, so what I was going to say is, referring to something Jordan had said earlier about as we learn how to manipulate the gut microbiota, it may not be that we just have one healthy state that we're targeting. It'll be if you're obese, we'll treat your microbiota to shift it towards this state that will help you lose weight. But then once you lose the weight, you might want to shift it somewhere else. Or perhaps you will develop colon cancer, and we want to shift it in a direction that will help prevent that from metastasizing. So there might be, I think what will happen is there'll be a future where, part of your doctor visit is going to be looking at your microbiota, looking at what diseases you might be facing, and shifting it to help to an optimal state for that condition for you at that time. So a sort of personalized healthy microbiota.
Yeah, and there's researchers working in California right now to have your smart toilet just tell you what microbes you might need in the morning. So, this could get real crazy real fast.
I think about a lot, if we're talking about our wildest dreams with probiotics, is could we slow down aging and age-related diseases as well? We can take the next question from the audience.
Hi there. I was curious. I had two questions. One of them, we refer to these all as microbes, but I know that's a pretty broad term. So are most microbes prokaryotic bacteria? Are there some fungi that live in our guts, or protozoans? And then my other question, the documentary kind of glazes over the explanation for it, but how is obesity linked to the microbiome? Like, how does having a stronger microbiome cause you, or a weaker microbiome cause you to produce fat more?
Yeah, so if you knew the answer to that last one, you'd probably be a rich person now. But there's hints at the original hypothesis was that there was these groups of bacteria, the firmicutes and the Bacteroides, and if you had the ratio of them out of whack, then that could help promote obesity. There's also things like, part of what the microbes are doing in our gut is they're fermenting these carbohydrates or proteins or things that they get access to. And some of those fermentation products, actually, we can take up as an energy source. So we get like something around 10% of our energy from these microbial metabolites. So in theory, if they're producing more of that, that might give you more calories per amount of food that you're taking in. At the same time, those products that provide the energy are also some of the ones that have the best health association data with them as well. So it's not fully understood, I would say.
Yeah, and to your question about the sort of the bacterial versus eukaryotic and viral, it depends, I suppose, if we count by mass or number. So I think if we go by number, viruses are actually the most abundant microbe in our bodies, but these are not viruses that affect humans, they're viruses that affect bacteria. Now, if we go by mass, it would be bacteria, because they're significantly larger than the viruses that affect us. And then, the eukaryotic component is there, so you're, but it's, orders of magnitude lower.
And then I'll give a little shout out for archaea as well. Those also are in your mouth and gut. So all sorts of weird, weird and wild critters.
Great question.
So I'm curious, you're using mouse models, and I know they have a very reduced microbiome in the gut, but it's a good place to start. Are there other animal models that people are working towards that might better reflect what's going on in a human body?
Yeah, the pig is probably among one of the best models for the human gastrointestinal tract. Without a doubt, as you can imagine, a pig is significantly larger than a mouse, which creates some logistical issues for those experiments. But the actual anatomy of their digestive system is much closer to humans than a mouse's is.
Yeah, and there are these notobiotic mice, so they've mentioned in there, and up here we've talked a little bit about it too. And when you have those notobiotic mice, you can put a human microbiome into them, so that you get a little bit. At least you're looking at the human-associated microbes in that case. Now, of course, there's still significant differences between their digestive tract and all of this as well, but you can get that kind of step closer.
I think it's also worth noting the microbe or the microbiome of mouse is actually not less complex than that of a human. It's just different, which creates challenges if we're trying to sort of translate what we learn about mouse microbes onto human microbes, because a lot of times there isn't a one-to-one mapping between those two things. So it's a little bit of an apples and oranges sometimes.
I was wondering, how do you define healthy microbiota? Is there a known community of good microbiomes that you define healthy, or do you look at the host that is healthy and think that that's healthy microbiota?
Yeah, I think we kind of touched on that earlier, is that it's really hard to define what is a healthy microbiota. There's certain bacteria that are often associated with health. So I mentioned some of these fermentation products like butyrate. So often you'll see butyrate producing bacteria being considered healthy. But there's a lot of other functions that the microbiota are doing, and quite a diversity of metabolic capabilities there. And probably, the best answer to what a healthy microbiota refers to the diversity of it. And so that you want to have as many of these potential metabolic functions as possible, with the hope that the ecology of the community is going to be more stable and produce all the beneficial compounds that you want. But really, it's almost impossible to define.
And very different depending on where you are in the world and what your lifestyle is. We can build, meta-analyses of thousands of people that live across the globe, and everybody who's industrialized is on one side of the plot, and everybody who lives, more traditional lifestyles and non-industrial lifestyles on the other side. And they're equally healthy. It's not that anybody's unhealthy, but they are very different, types of microbiomes. So it's really, again, relational to your environment and your diet and your lifestyle.
Next question from the audience.
I can say, oh, thank you.
So my question was related more towards the ethics of some of the human trials. So we talked about like using PEG or antibiotics to clear the microbiome before, for example, like an FMT. So in those specific cases, or even for other studies where you're using like PEGs or antibiotics to clear the microbiome, what are like the current ethical considerations behind that? And are there certain ethical considerations kind of pushing studies towards using more patients in more dire circumstances, and would that affect the results of some of these studies?
Yeah, I mean, many people who are enrolling in these studies today are people who are suffering from a current ailment, right? And so you wouldn't just a priori go and ask somebody to clear out their microbiome and see if you can just shift it without there being some impact. And so you have to be, very careful about comparing. Like in our studies, we'll compare everyone who has necessarily disease, but you'll compare, for example, the standard of care as the gold standard to, say, a fecal microbiome transplant. The classic example of this was actually done in C. diff infections, where they were comparing patients who had C. diff with antibiotics. They were giving them antibiotics, and then they were comparing patients who had C. diff who were getting fecal microbiome transplants. They actually stopped that clinical trial early because it became unethical to continue giving people antibiotics because the fecal microbiome transplants were working so well by comparison. So you have to be very careful, obviously, about long-term harms when you're doing these sort of studies, and it's why it took the FDA a long time to approve FMTs, even in the context of one disease, a C. diff infection. And they're still not approved by the FDA for many other maladies and treatments. They're still considered experimental treatment at this stage, and there are significant risks that are carried along with that for people enrolling in those studies. So it's not something we take mildly, but it is interesting to think about how enrollment in those studies has changed over time and how it may continue to change, especially I think as awareness of FMT and its again potential for disease cures and how people view that and whether or not they'd want that or not.
And part of doing human research is, it has to be approved by an ethical board, which is, usually accredited by, a higher governmental agency. And part of that is, essentially convincing other scientists that this has a plausible reason to work and that, the potential benefits outweigh any risks associated with it. So if you said, I want to do fecal transplants to make my hair grow slower, right? That's probably not going to get approved because A, why, and B, maybe the risk benefit doesn't quite work out on that. So that's sort of a component of it as well.
Yeah, and so by the same, so that risk benefit part is crucial to it. So if I'm telling you that I want to just change your diet slightly to make your hair grow slower, that's a lot more likely to get approved than blasting you with antibiotics and giving you an FMT to do that. It may not be a huge benefit, but there's also almost no risk associated with it as well. And then just a little note on the ethics, that even for animal trials, there's ethical considerations that go into it. How many mice are you using? How sick are you going to make these mice? That sort of thing has to be considered as well.
Hi, I had a related question. So I know we talked at length about what's a healthy microbiome and about FMT, but when doctors give these fecal microbiota, they must have gotten it from somewhere, and they must have a decent idea of what it is. So, hopefully, so what is the criteria on picking the donor for FMT, and do all recipients get the same community, and how is that controlled, and so on?
I was just going to say, if you look in that video that we saw, so the woman that was talking about receiving it, she got it from her daughter, right? And so a lot of times it's historically been what's a convenient, healthy donor that you can find that's willing to give it to you to do this with. I think it's moving towards becoming more intentional and screening for the presence of pathogens in the sample and things like that. But yeah.
So in the FMT trial, in the video, we actually saw some containers that had an open biome label on them, which was in fact an organization that was trying to distribute fecal material for clinicians and for research studies. Part of that was, the individual would have to be, healthy, lean. They'd be screened for a number of pathogens and parasites. Unfortunately, there were also instances where pathogens slipped through not being detected that caused problems. And then, this kind of goes back to the idea that there is risk associated with FMT. But, oftentimes it would come from somewhere like open biome or, a healthy relative.
Yeah, many of the initial studies didn't even necessarily characterize the donors when they were sort of going through the study. And so after some of the very successful FMT studies were completed, they then had to go, okay, wait, but what was in the donors? And go back to the donors and then recharacterize them. And so it created a little bit of a lag in the field to really say, okay, this is what makes a good donor. And we're still sort of figuring that out. And there's initiatives, like the one that Jordan just mentioned, but also ones in Australia that are biobanking all sorts of donor material and trying both single donors as well as mixed donors, and mixing up to sort of 10 donors all at a time in hopes that will be more beneficial. And I think I don't know, the jury's still out as to whether or not mixing donors is better or worse. But it's an active area of research. But I think as we're developing oral microbiome transplantation, we're thinking very hard about how do we select those donors, and then how do we develop new tools to be able to screen those donors and really poke and prod them, in a safe environment, not in a human, to really then understand whether or not they're likely to be efficacious and not cause harm to the person.
And this idea of every donor has their own microbiome. So even within a study, it's like every patient's getting a different drug. And so you could almost think of a fecal transplant like going to the pharmacy. They grab a whole 100 random pill bottles, mix it all together into one thing, and give it to you. And then the next person comes in, they got another 100 random drugs mixed together and given to you, right? So I think that is a fair analogy of how FMTs works. And so we do need ways to standardize them. And that's being done commercially now, relating to C. diff and standardized treatments that try to homogenize what's actually being given. Yeah. But, this is again where I think in the future, these sort of synthetic alternatives to fecal transplants are going to have to become more prevalent.
Yeah. If you need some reading tonight, there are some pretty crazy articles about people who are routine fecal microbiome transplant donors, and then every week on Monday they'll just go drop off their feces. And so some people are known to be really good donors, and they may not know why they're a good donor, but people know they're a good donor, and so they will be, regularly sort of providing that. But it's all moving, I think, to sort of commercialized products. But definitely in the past there were some routine donors who were highly sought after.
I actually have a follow-up question to this. So in the movie, they highlighted that there was an ancient medical practice of giving fecal matter and urine to people. And so what can we learn about ancient medicine before they had antibiotics? And are people looking to that for inspiration for current research?
Yeah, absolutely. And that's not necessarily an ancient practice. Some cultures will still do things very akin to that of giving people feces in order to restore microorganisms. This is also widespread in the animal kingdom. Thinking about coprophagy, koala mothers feed their feces to baby koalas so that they can get the right microbes that can protect them against toxins in eucalypt trees. And so it's not a new idea. It's just a matter of commercializing it and making it a modern Western treatment that's a new idea. And so certainly, the Chinese were not the only ones to have this sort of history of doing this, but it's something that I think we're actually realizing there was there was something to feeding someone a dragon labeled stew. Yeah.
I would have loved to have been there the first time someone suggested it.
Yeah. Or just the smell of it. Yeah.
Next question.
Hi. So I had a question about the organism's effect on kind of how the microbes are competing against each other, and if it was more of like an arena of just the microbes, or if the human is having some kind of control over who's winning and who's losing.
And you're referring to in an FMT?
No, not necessarily. Just like in daily life.
Okay.
Are there things that maybe genetically some bodies are able to better facilitate a helpful competition than others?
Yes. So, there's a reason why, the types of community experiments done in a test tube don't work inside a mammalian body, right? Because the host is a huge part of it. The host is, through the immune system selecting against certain microbes, it's feeding certain nutrients, taking others away. It's a big component of what's shaping the community. without a doubt. And that's actually part of what makes it so complicated, right? Because we're considering a network of hundreds and thousands of microbes interacting with each other, plus with the host, and very quickly a lot of the ways we think about, you know, ecology and the mathematical models fall apart completely and are borderline useless. And I think that's where a lot of the really exciting advances are coming more from using machine learning and deep learning, these approaches that we see in, you know, data science and Silicon Valley, moving into the life sciences to help address some of these complex problems, which, you know, our existing tools just don't work for.
Yeah, I mean, there are several studies at this stage which showed relationships between, say, for example, your genotype or different variants or SNPs within your human genome and how that correlates to particular microbes. And many of them are within immune genes and ways that we know the human body interacts with these microbes. But again, like Jordan said, the ecological models for understanding this complex interaction between all of the chemical processes in the human body versus all the microbes and their chemical processes fall over. And we don't really have the tools yet, I think, to broadly understand it.
And just to point out that pretty much any ecological concept that you can see in the macrobe world also exists in the microbial world. And so it's not just about competition and things like that. There's also cooperation taking place as well. And, you know, one bacteria might be producing a waste product that is now the food source for another bacteria in there. Or one bacteria might have some of the enzymes to break down a substrate that's coming in, but needs the help of one or two others to fully break it down and get access to all those nutrients. So it's a very complex interplay happening.
So along those lines, at the end of the movie, they mentioned fecal transplant pills. Is that what it sounds like?
Pretty much, yeah.
Usually a fecal sample would be freeze-dried and then put into a capsule, and that's what those would be.
They were called poop pills, I think, in most of the headlines I saw, but they largely, again, have not been effective, is my understanding, because of the problems that Daryl are talking about. Those microbes are going to struggle to get established in somebody who already has existing microbes in their gut.
Koalas have figured it out, but we haven't figured it out yet.
Right. Or you need to do those transplants in very young children, which raises a whole bunch of very new ethical issues, which have not yet been been discussed.
Has your research affected your lifestyle or diet?
You know, I always ask myself if I had enough fiber today. I think that's a big component because that is a major energy source for our microbes. But not all fiber is created equally, and I think that's an important component. I mean, really, at the end of the day, fiber are just often, you know, carbohydrates that we can't digest, so they get past us absorbing them and get to the microbes. But, you know, something like, you know, psyllium fiber might be detrimental, but some other like common fiber supplements might actually be deleterious or bad for you, or in certain ways pertaining to liver health. But, you know, rather than, you know, go after sort of fiber supplements, I think the key is really to look at diet and make sure that the diet itself is diverse as possible, because if your diet is as diverse as possible, so too will be the nutrient sources getting to your microbiome, which leads to a very diverse microbiome. And there was actually a manuscript a couple years ago related to this big project called the American Gut Project. And I think the answer was 28 plant products a day.
Yeah, 30 is the number 30? I have in my head, yeah.
So eat 30 plant products a day, but I think pepper counts as one. So, you know, a little liberal on what's a plant product.
Yeah, that number is definitely in my head every day, and it's annoying because I can't get it out of my head. It's like, have I had 30 plants this week, you know? And so, you know, and there's easy ways to cheat and get there, you know, by these, for example, the breads that have 21 you know, different grains or oatmeals or things. But, you know, you find yourself calculating these things in your head when you don't probably need to, but they stick with you.
Yeah, and I'll also point it out, like, when I'm trying to raise my children, and, you know, often fiber is like one of the hardest things to, you know, get them to have good amounts of it, especially diverse food sources of fiber. And, you know, I'll second the point about it definitely being better to be getting those from a diverse set of foods rather than trying to, you know, chase after the ultimate supplement that is going to benefit you. You know, we do trials with supplements because, as I said earlier, you know, it's our kind of way of cheating of getting lots of that fiber into the system. But probably that's not actually what you ultimately want to do. If we feed a ton of that one type of fiber to a person, yeah, we might see that increase in that butyrate, like I was talking about. But the other thing that'll happen is a decrease in diversity of the community, because we'll select for the bacteria that are really good at using that fiber. And so ultimately, what you'd really like to have is a wide variety of fibers to feed a wide variety of bacteria.
As a follow-up question, so I've recently started making my own kimchi, and you hear a lot about fermented foods and how they're good to include in your diet. Is there any scientific evidence to support that?
Yeah, it's an interesting question because I think, as Daryl said earlier, many of the probiotics that are in some of the commercialized food sources that we have, either the food source is pasteurized, so you don't get actually live organisms, or that microorganism will not ever necessarily become established in your gut because it was designed to ferment milk and make yogurt or whatever that looks like, right? But there is, again, it's complicated, I would say, research, and it's been marred by conflicts of interest with companies trying to sell probiotics. But there is some evidence to suggest that some of these probiotic organisms have anti-inflammatory effects. Again, not from the living probably constituents of the microbe, but you know, something that they have in with them. So I think about that a lot in my daily life and have started fermenting a lot of food so that we can eat naturally fermented foods that don't just have the 5 industrial strains, but actually have strains from our garden and from the environment around us, much more akin to sort of a pre-industrialized lifestyle. So I also do a lot of gardening as well to kind of add on to your kimchi, but it's those things, thinking about natural sources of microbes, that I think a lot about.
Yeah, you know, the real studies, like the significant studies about fermented foods, are actually quite limited, and there's a lot less of them than you would probably think. But you know, there's a big component of that. There's a commercial standpoint about what's proprietary, but the problem is, you know, your kimchi won't be the same microbes as my kimchi, which might not be the same as Laura's kimchi, right? And we go back to this idea is that might be a false comparison because they all have microbes, but different microbes, and do we expect all microbes to function in the exact same way? Maybe not, and so that creates a real problem in how we actually do those studies.
Yeah, because you'll see things like these sourdough projects, and you know, people will kind of transfer their sourdough starter cultures around. But even when you take the same one and transfer it over time, that culture will change. Let alone if someone from a totally different part of the world had a sourdough culture that they were using. And so it's the same with kombucha and kimchi and all these other things too. You know, probably they're not doing any harm, I wouldn't think. And there's also this idea of, you know, you probably heard of probiotics and maybe prebiotics, but we're also getting now postbiotics. So some of the products that bacteria are producing in our gut, can we just take them directly? And to a certain extent, that's what you're getting in those fermented foods as well. So, but there definitely needs to be a lot more research on it.
I think Laura hit on a very important concept here. You know, when it pertains to a lot of these foods that would have used to contain a ton of microbes are pasteurized or sterilized. And so sometimes the fermented foods you might get at a grocery store aren't really what you think they are, right? And I think kombucha is a great example of this. So, you know, real kombucha is made from this very complex community. And if I can be honest, in my opinion, it tastes absolutely horrible. And the reason it tastes horrible are all those compounds that should be good for you. Now, the problem with what you find in a store is it's almost what they've done is they've almost made a soda or a pop, however it's said in Pennsylvania. And then what they'll do is they will add things like cider vinegar that sort of mimic the flavor of the natural fermentation. They'll add organisms in after the fact, like some of these lactobacillus or bacillus spores. But that itself, from both, you know, a chemical perspective and a microbiological perspective, is nothing remotely close to what kombucha is if it's actually made at home. And so that's something you really have to be thinking about if you're going to be buying these sort of products with the intent of, you know, I want to get microbes in my diet.
Yeah. I think just to add on to that, the other way I think about my microbiome on a regular basis is just also my environmental exposures. So there's some really amazing research looking at soil microorganisms, specifically Mycobacterium vaccae, and how it may have positive benefits on your mental health. And so I think about that a lot when I'm, again, outside, digging in the soil, interacting in diverse ecological environments. We've also done studies on kids and looking at whether or not they get more microbes from interacting in forest-based environments versus just a green, grassy field versus in the classroom. And of course, no surprise, they get more microbes in their bodies and on their skins by playing in diverse forests. And so I hear my son go, yes! But really, it's from my perspective, it's thinking about what are the environments in which I interact, in which I spend most of my time. Am I really just walking on concrete and sitting in my office all the time, or am I getting out and getting in some of those diverse environments and interacting with those environments in ways that could be meaningful to my microbiome?
That's a great point. And I would also highlight that the foods that we eat, those plants have their own microbiomes that originate from the soil microbiomes, and that there isn't a lot of knowledge on how different plant microbiomes then impact our microbiome. So it's all really connected. Take the next question.
I wanted to say that I am taking guilt away from this after watching this because of taking my child to, when she was young, for strep infections and having the antibiotics many times. Now is gluten sensitive and very anxious a lot of the times. And another comment I wanted to make is my husband many years ago ran a daycare center, and the healthier kids were the ones that picked the gum on the bottom of the chairs and chewed all the gum that they found. The other question that I did have is, was there any research done on the blue zones, the microbiomes of the people that are living there because of the health and the longevity? If they're a healthier group of people, why are they included in any of the testing?
Yeah, so there has been at least one study that I'm aware of on the blue zones, and folks from those areas do have an increased diversity, meaning they have more different types of microorganisms, especially into older age. We see generally a decline in your gastrointestinal diversity as you age. And so maintaining that diversity appears to be an important factor, although, it's unclear, I think, in my mind as to whether or not that's something physiologically controlled by our human body, or whether or not that's because people don't go outside as much or whatever, or change their diet when they get older. But I will also say, you shouldn't, as a parent, walk away with a bunch of guilt because, things like strep infections in your throat can be very life-threatening. They can still be very, very destructive and very evil. And so there are times antibiotics still definitely need to be used. It's just finding, like they said in the movie, better ways to figure out when to use them and how often to use them. And so, without what the generation's gone through, we wouldn't know what we know today. And we wouldn't have the information to try to undo that. So antibiotics still are a very crucial part of our medical repertoire, and they're not all bad, even though, we're learning how to use them and when's appropriate.
Yeah, definitely. I think it's a great point that, probably all things that, we're doing, raising our kids today, we're going to find out how many of them were totally wrong in a few years. I think that's, pretty much every generation goes through that. So, and then, the point about the antibiotics is, don't be afraid of antibiotics. They're like the greatest medical invention ever. So, if your doctor if your doctor wants to give you antibiotics, bring up your concerns about, your microbiome and whether it's truly necessary. But if they say you need to take these antibiotics, you probably need to take those antibiotics. So,
one thing I would bring up that I know about the blue zones, or these people who live a very long time in those regions, is that something that's similar across these blue regions is that they have very low stress, or they live low stress lives. And we haven't touched on that at all, but how can stress impact your microbiome?
So I'll come at it from the mouth because we spend a lot of time thinking about the microbes that live on your teeth. And there's a really amazing study that came out last year showing that the microbes in your mouth actually respond to cortisol directly, which is a stress hormone. So it's not just your body responding to cortisol, it's also the microbes in your body directly responding to cortisol. So these microbes have, evolved the same adapters and similar responses to these stress hormones that are released in an environment where they've lived for millions of years. And so absolutely we have to assume an effect, on these microbes, but I would say how we modulate that effect or what that does in response, when the microbes are responding, what does that then do as a secondary effect on the human body? We still, really don't know yet, in the mouth anyway.
Yeah, I think one thing that comes to mind in the gut is, stress's impact on like gut motility and things like that. And sort of the rate that things are passing through our gut is going to affect our microbial populations that are there. Like how long do they have to work on those fibers that we put in there? And so, they're difficult to digest things. If they don't have enough time, they may not be able to get all the benefits out of it for you.
And I think, something to keep in mind is, in the human body, we always think of all these systems being separate, but in reality, everything's tied together, right? And so cortisol can have wide-ranging effects all over the body, including on, GI health and microbes. And, part of, I think, what we're trying to do, scientists is really try to understand, well, what's actually happening? Can we really distill that down to the function? And then once we understand how it works, can we manipulate it? And that's actually why I think the microbiome is such a great place for young scientists to go into, because if I may be so bold, I think there's still so many important discoveries left to be made. And so, the potential right now is, almost limitless, I think.
Yeah, we talk a lot about microbial redundancy, right? And that there's probably also, even if one person finds one microbe that does it over here, there might be another microbe over here that does it maybe slightly differently. And so, even things that are discoverable are rediscoverable, I think, in microbiome science, which is really exciting.
So we're actually approaching our time limit here. It's almost 9 o'clock. But I just wanted to present one more question that was submitted online. Someone asked, is there any way for community members to get involved with research or other microbiome center efforts at Penn State? So can you speak to that?
Yeah, so you will often see advertisements for people conducting clinical trials on microbiomes. So the official sort of Penn State distribution for that is called Study Finder, and you can look on that to see if there's any microbiome studies ongoing that maybe you could be participating in. And you might see them advertised around different places as well, and they range in what is required of you. Some of them it's just eating something for a little while and providing a fecal sample. Others it might be more involved, blood draws and things like that. But if you're interested, there's ways for sure.
And I think too, if there's something that really interests you, you can always email. You can always reach out. Like I remember when I was in grad school, I had a conversation with a farmer from Alberta who was concerned about glyphosate pesticide affecting the microbiome. And I didn't do anything about it at that time, which was kind of a bad choice because it turns out that's a very interesting area, right? But I think you would be surprised how willing people are to talk about things, right? And so you can always reach out, I think, to anyone with ideas or ask questions. And I think most of the time, plus or minus a couple of days on the email, most people would be really happy to talk about what they're doing or sort of your ideas.
Yeah, and I'll just say we put up our events and outreach events like this up on the Microbiome Center website, which is found in the Huck Institute's of Life Sciences website. And we love interacting with the public. It's honestly my favorite part of the job is talking with people about the science that we do.
Yeah, and to add to that, at the center level, we're hoping to connect with the greater community a lot more. And again, if you do have ideas and you want to reach out, the center is a great place to help get you connected to specific scientists who have similar interests as you. So I guess that concludes our event. Can we give one last round of applause for our amazing scientists? And thank you for your time. Good night.